Uh, my students, today I'm going to speak something about transgenic plants and their applications in crop improvement and the limitations. Uh, so transfer of a gene, transgenics are nothing but the transfer of a gene from an organism into a plant cell and integration into the genome material or a genetic material of that later, usually employing a recombinant DNA technique, is known as a engineering of the plants. So these are nothing but those plants which have been genetically engineered are transgenic plants. So it's a most potent biological approach which constitutes transfer of a gene assembly, specifically constructed gene assembly through various transformation techniques. So previously you have learned about what are the different kinds of transformation techniques like direct gene transfer methods like gene gun or a bialistic method, electroporation and all, or indirect method by using agrobacterium mediated gene transfer and all. Then the plants obtained through genetically engineering contain a gene or a genes usually from unrelated organisms, so which are unrelated, hence we call them as transgenes, and the plants containing those are called as transgenic plants. The first transgenic plant was produced in 1983 when a tobacco line expressing kanamycin, which is an antibiotic resistance, was being produced. Soon after, transgenics have been produced in many plants for which are resistant for herbicides, insects, viruses, or sometimes expressing male sterility, especially in the case of Barney's and bar star systems. Delayed ripening or slow fruit softening were being developed. You know much better. I have already told this example in the previous classes. The flavor saver tomato, which was regarded as the first transgenic variety to reach the market, which has a prolonged shelf life, it remains fresh for a prolonged period of time. Then the role of transgenics in crop improvement. So these transgenic plants carry some additional foreign genes which are transferred from unrelated organisms. So the combined use of recombinant DNA technology, gene transfer methods, and tissue culture techniques have led to efficient productions of transgenics in a variety of crop plants. So these transgenics, which are also called as genetically modified plants, GM crops, have both basic and applied uses. So we quickly look at in what ways they have been useful. So they are increasing the efficiency of the crop production systems. So for instance, the transgenics resistant to herbicides, insects, viruses, biotic and abiotic stresses have been developed or produced. Then transgenic plants is an effective means of inducing a male sterility in the croplands. Example, I have told you, Barney's bar star system in the Brassica napus. And then transgenics are also used for stable producing of some kind of things in food processing industry. The best example, bruise resistant and delayed ripening in case of tomato. Then several gene transfers are aiming at improving the produce quality. Say for example, protein and lipid quality can be improved. This can be achieved either by suppression or overproduction of some kind of exogenous genes. So we are trying to suppress some gene or we are trying to encourage that gene. So that happens. Then transgenic plants are aimed to produce novel biochemicals such as interferon, insulin, immunoglobulin, etc. Useful biopolymers like polyhydroxybutyrate, which are not produced by normal plants. So these are compounds extracted from the plants, can be used by the used as a pharmaceuticals or industrial substances. This can be done. Then utilizing these transgenic plants as a bioreactors or the factories for production of the specific chemicals and the pharmaceuticals is referred to as molecular farming. So just like agricultural farming, these molecules are being produced using the plants as a bioreactors or the factories. We call it as molecular farming. Then transgenic plants have been produced expresses a gene encoding antigenic protein from a pathogen. So use of these can be used in case of vaccines for immunization against pathogens has been fastly emerging as an important object. For the information of you, Now currently for the COVID-19 also, they are using plants for development of the vaccinations, because when we are testing it on the plants, the plant dies, has no loss. When we are testing those directly on the humans or the animals, it is a little problematic thing. Hence, transgenic breeding has a lot of scope, especially today for COVID-19 also, for development of the vaccinations. They are injecting different kind of antibodies or antigenic proteins into tobacco plants and trying to find out the results, how they are showing. Let's see how they are going to be successful, to what extent. Then, transgenic plants have also been proved to extremely valuable tools in studies on plant molecular biology, regulation of gene action, identification of regulatory, promotory sequences, etc. And what are the limitations associated with this? So, this exhibits unstable performance. So, we are aiming for some kind of goal, and we have engineered that plant for that goal. Say, for example, production of some kind of biomolecule. We have engineered or we have transferred a gene and we are trying to express it. But what we are expecting was not shown sometimes, or it shows uh, unstable results. So these are the problems. Then transformants have undesirable side effects. So you are adding something, you are getting in return additional unnecessary things along with it. So this is also one problem with the transgenes. Then the position and integration of the foreign gene in a host genome affects the expression of the transgene. So you insert your gene of interest at a particular point, or as when you call a location in the host genome, it expresses in some way, and when it is inserted at a different position, the expression is entirely different. So we cannot get the exact what do you call uh, location. We cannot go for the precision in the location of the gene transfer till to date. Then, however, we are using with the genome editing technologies these days. That's a different issue. But transgenics, it's not possible. Then, inability to transfer the polygenic traits. So those traits which are governed by many genes, we cannot transfer them as such, because those are governed by many genes. You need to transfer a group of genes together, and those group of genes are spread over a genome to an extent. So you need to identify them and you have to transfer them. At a stretch, it's not possible. Then transgenic breeding is a very expensive method, very expensive really. Then it requires high technical skills, and transgenics are recovered at a very low frequency in the cell culture, and moreover regeneration of the transgenic cells into the whole plants is also difficult, and it's a time consuming task. And transgenic breeding acts against natural evolution. That's a limitation really. So it is these transgenic breeding and natural evolution are two opposites. These are contradictory terms. Then there are chances for development of new weed species through transgenic breeding. Say, for example, we are trying to produce a wheat herbicide-resistant kind of thing. Then periodically we are doing this herbicide sprays, and then some of the weeds evolve themselves and they become very noxious. And then you have very new weed species into this world, which cannot be controlled with herbicides either. And sometimes the foreign genes have adverse effects on the genome of the recipient parent. In such cases, it may give rise to useless gene components, which may become a problem. So this last limitation has been always put forward in stepping down the transgenics, especially for foods or a food growing, what you call a consumable crops in case of India and also many of the European countries. Then engineered for insect resistance, herbicide resistance, and disease resistance. So, rTNA technology, gene transfer methods, and tissue culture techniques are integrated in this transgenic technology. Then, the most important thing is the most major global area under transgenics under the soybean crop, which is a very important thing to understand. We may be thinking soybean is not seen in India. To that extent, yes, transgenic soybean is not accepted because it is an edible crop and it's not accepted in India. But the transgenics across the world, soybean is the major area grown under transgenics. It's about more than half of the world's soybean area is cultivated with the transgenics. Then today, research and development efforts are targeting for resistant plants for insects, herbicides, and diseases. Excuse me. Then engineered insect resistance. So there are two approaches for developing insect resistance. Number one, introduction of the resistant genes from microorganisms, and two, from the higher plants. So from resistant genes from microorganisms. So the cry gene, which is very well known for you, that is Bacillus thuringiensis, produces a protein which forms a crystalline inclusions in the bacterial spores. These crystal proteins are responsible for insecticidal activities of the bacterial strains. So these insecticidal activities have been classified into, and they are grouped. So cry1, cry2A, cry2B, cry3, something like that. So cry1, cry2, cry3, and cry4 proteins are parasporal inclusions, the crystal proteins from the Bacillus thuringiensis that exhibits hemolytic activity. So these proteins are solubilized in the alkaline environment of the insect midgut, and these proteolytically processed to yield the toxin core fragment out of the bacteria, which are going to kill the larva of the insect. So the toxic function is localized in the N-terminal half of the protein, and the C-terminal of these proteins is highly conserved and involved in crystal formation. So cry1 proteins are insecticidal for lepidopterans, cry2A, Lepidoptera and Diptera, cry3B, Diptera, cry3 Coleoptera, cry4 Diptera, cry5 and cry6 for nematodes. These are insecticidal. So the toxic action, how it's going to happen. So when the cry proteins are ingested by the insect, they are dissolved in the alkaline juices present in the midgut or the lumen, that's the digestive tract. The gut protease its digesting proteins, process them hydrolytically to release the core toxic fragments into it. So the BT, a lepidopteran specific gene, especially Bacillus thuringiensis of subspecies Karstaki, has been widely and successfully used in tobacco, tomato, potato, cotton, rice, maize for developing resistance for Lepidoptera pests. Then a lowered level of expression of BT gene, they have intensified through the choice of the suitable promoters or by modifying the coding region of the BT gene. So especially by using 2 copies of the constitutive 35S promoter, which is from cauliflower mosaic virus, which was been derived, has been shown to provide a very good result. So 2 copies, in the sense we are trying to put it in 2 copies, just like you make a 2 copy of Xerox, something like that. You put 2 copies of promoter sequence. So you know the operon concept very well, I hope. So promoter, operator, region, regulatory gene sequence, how it is, that's the same thing, functions everywhere. And the use of redesigned synthetic BT gene with increased guanine and cytosine content have also been used in some of these crops, and several instances that synthetic version exhibited up to 500-fold of increased expression. So that's how it is going to trigger. So it is more toxic 500 times because of these redesigning techniques. Then, so the best examples of boll gourd cotton, yield gourd of maize, new leaf of potato have been released, which are all resistant transgenic varieties. The great diversity of delta endotoxin coding genes, each of which specifies a spectrum activity of, for example, cry1AB protein is highly toxic for European corn borer, which is used in hybrid BT corn hybrids, and cry1AC for tobacco budworm and cotton bollworm, and cry3A especially in case of potato for Colorado potato beetles. So this is how, so when the larva ingest the BT leaf or something like that, so it goes inside. Actually that bacterial strains are having a crystalline protein. This is a spore, So crystal protein, when it is entering into dissolution of the protein crystal happens here because of the proteases of the gut, then the bacterial spores and also the crystalline proteins are released. So activation of the protein happens, the crystal which gives rise to delta toxin and finally toxin. This toxin binds to specific receptors of the peritrophic membrane, and then vacuolation of the cytoplasm, finally cell rupture, disruption happens. So this is how the gut is totally collapsed of that insect and finally dies after feeding this bt cotton, something like that. Then introduction of the resistant genes from the higher plants. So from the higher plants also you can take it, the gene transfer activities. So protease inhibitors, the cowpea trypsin inhibitor gene has been transferred from cowpea, which produces so anti-metabolite substances, so which is dangerous for storage pest, that is a bruchid beetle, uh, the Callosobruchus maculatus. So it will, what it does is it inhibits the protease function. So when it feeds on the protein, if there is no protease, so protease is not there. So what happens? Protein doesn't get digested. So then, so what if it is not get digested? It cannot grow. So that will result in retarded insect growth and development when ingested in high doses. Then alpha amylase inhibitors. So this gene is isolated from Fasciolus vulgaris and is expressed in tobacco, that inhibits the insect gut alpha amylase. So when larva generally secretes a gut enzymes, that is amylase, that digests the starch, by adding this protein that inhibits the amylase, then insect starves and it dies. It don't get food. Though it consumes starch, it cannot convert it into a useful thing because of the lack of amylase. Then lectins. So these are the plant glycoproteins which are used as insect toxins. So lectin from snowdrop, that is Galanthus nivalis, is known as GNA because of whose activity is insecticidal for aphids. And then engineering for herbicide resistance. So here the major role is they either inhibit the photosynthesis or the biosynthesis of essential amino acids. There are two approaches for herbicide resistance. The first one is detoxification. So introduction of a bar gene cloned from bacteria, that is Streptomyces hygroscopius, into the plants makes them resistant to herbicides which are based on phosphenothricin. So this is an active chemical, phosphenothricin. So phosphenothricin based herbicides, when bar gene is added from the clone from the bacteria and it is incorporated into the plants, they become resistant. So the PPT herbicide kills the plants by inhibiting what? It is a glutamine synthase enzyme. So glutamine synthase is inhibited, because of which the plant dies. Then the bar gene produces an enzyme, phosphenothricin acetyl transferase (PAT), that degrades the phosphenothricin into a non-toxic acylated form. So it breaks down the phosphenothricin because of producing. So bar gene encodes for what? It is phosphenothricin acetyl transferase (PAT) enzyme is produced from the bar gene. So when we are incorporating bar gene into it, it will produce PAT. So PAT is going to fight with PPT and it is going to dissolve it into a non-toxic acetylated form. So the plants engineered with bar gene were found to grow in PPT at levels of 4 to 10 times higher than the normal field application. So up to that level, these plants which are having bar gene are going to resist the herbicide application. And likewise, we have BXN gene from Klebsiella ozonea, which produces an nitrilase enzyme that imparts resistance against bromoxanil. And also BXN cotton resistant varieties have been released in already released in USA. So Streptomyces, what we are doing, we have isolated bar gene which is encoding for phosphenothricin acetyl transferase PAT, that produced enzyme PAT, and PAT is going to detoxify phosphenothricin into a non-toxic form. So it doesn't get affected. So if you observe the before application, the plants look like this, after application like this. So finally again gets recovered normal plants, and you observe this. These are our plants, these are our intercultivation area where weeds are removed with the help of herbicide application. So then target modification, this is the second system of herbicide. So transfer of the gene whose enzyme product becomes insensitive to herbicide. So in this system, a mutated gene is introduced and that produces a mutated enzyme in the plant that is not recognized by the herbicide. Hence, it doesn't kill. So you may observe some movies like that. So it may be a monotonous lecture. I will show you. So you find some movies, the person who is trying to escape, he just changes his dress so that the people who are trying to find out him don't look for him because he has changed dress, because they are behind his dress. So in this way, here also what we are doing is we are trying to put a mutated gene, introduce a mutated gene that produces a modified enzyme, so which is not recognized by the herbicide, and that's not going to kill it. So for instance, the mutant AROA gene from bacteria Salmonella typhimurium has been developed for developing tolerance to herbicide glyphosate. So the target site of glyphosate is chloroplast enzyme 5-enol pyruvyl cyclic acid 3-phosphate synthase, EPSPS. It's shortly called as EPSPS. So inhibiting the synthesis of aromatic amino acids. So introduction of mutant AROA gene produces modified EPSPS. So our target site of glyphosate is EPSPS, and this mutant gene produces modified EPSPS, so which is not recognized by the glyphosate, and hence it provides herbicide resistance. The best example for that is Roundup Ready soybean, which is providing resistance to glyphosate. So likewise, we have also developed a herbicide resistance mechanism for different herbicides like sulfonyl urea, imidazolines. Herbicides inhibits ALS, chloroplast protein, which inhibits the synthesis of leucine, lysine, valine. Tolerance to these herbicides have been achieved by engineering the expression of ALS-derived plants. Then engineering for disease resistance. First of all, we'll read about virus resistance. So disease resistance generated by employing pathogen genes, which is referred as pathogen-derived resistance. These are again two types. The one is a coat protein mediated resistance, CPMR. So in this, what we are going to do is we are trying to introduce a viral coat protein gene into the plant and make it resistant to the virus from which the gene and the coat protein were being derived. So if a susceptible strain of crop is inoculated with a mild strain of virus, then the susceptible strain develops the resistance and against the more virulent strain, which is normally done in case of antibiotics or the vaccinations. So Powell, Abel et al. Have first demonstrated this with the TMV coat protein that showed resistance similar to that occurring in the viral mediated cross protection. So the accumulation of their protein in unaffected cells results in effective resistance by uncoating the virus particles before translation and replication. So virus, when entered into it, it will replicate, and also protein synthesis happens. Transcription, translation. So replication, transcription, translation happens, and all viral particles are going to release. So they are going to stop those. So it was first demonstrated in TMV. So in the first picture, if you observe, which is a non-transgenic infected cell. So when virus gets infected, what happens? You see the protein coat is separated, and the genetic material has entered into the bacterial cell, and it further what happening? Replication is happening, transcription is happening, and so replication is happening first of all. Then virus protein, coat protein, it's also what is happening here. Protein transcription, translation is happening, and now the replication also is ready. This genetic material again enters into the newly produced coat proteins, and in this how the bacterial cell entirely is being colonized. So in the next picture, if you observe, the transgene expressing coat protein has been already inserted into it. Now what has happened? So the capsin protein is produced, and now here what happens? So the gradually the this coat protein is already made in this. So what is happening? Whenever the virus is entering into it, it is engulfed, and coat protein will be surrounded by it, and it is left out. So the transgene expressing a coat protein, but no non-structural proteins are produced further. How? Because mutant coat proteins are going to suppress these things, and there is no non-structural proteins produced thereafter. So in this how resistance is given. Then satellite RNA mediated resistance. So some RNA viruses have small RNA molecules which are 300 nucleotides in length, which are called as satellite RNA. And these satellite RNAs are RNA molecules dependent on the helper viruses for their replication and transmission, even though they are unrelated to viral genome. The presence of them leads to reduction in the severity of disease symptoms, and they have been used to develop resistance against specific viruses. The best example is cucumber mosaic virus resistance with the help of satellite RNA. Then fungal. So fungal antifungal protein mediated resistance. So here chitinase and glucanase encoding genes are being used. So what they are going to do? So whenever chitinase is which degrades the chitin, then the fungal cell is made up of chitin and glucan. When these chitinase and glucanase are there in the plants, they will break down the cell and hence providing resistance. So when they are together, it is giving more resistance in case of fungal. Then antifungal compound mediated resistance. So the low molecular weight compounds such as phytoalexins possess antimicrobial properties and they are providing resistance. The expression of the stilbene synthase gene from grapevine in tobacco resulted in production of new phytoalexin compound, that is resveratrol, and this has been enhanced the resistance to botrytis cinerea and blast resistance in rice. Then active oxygen species AOS, including hydrogen peroxide, they play a role in defense mechanisms especially. So transgenic plants which expressing H2O2 generating fungal gene for glucose oxidase was found to have elevated H2O2 levels that have provided resistance for verticillium wilt. And bacterial resistance, bacteriophages sick T4 lysozyme in transgenic potato tubers have led to resistance in Erwinia keratova. And similarly, barley alpha thionin gene significantly enhanced resistance of transgenic tobacco to bacteria Pseudomonas syringae. So here are the list of insecticide, herbicide, and virus-resistant transgenic varieties approved for commercial cultivation in USA. So if you observe boll gourd cotton, that is BG cotton, BT cotton we call, it was approved in 1995 firstly by the company named Monsanto, first time in the USA, and also bromexidine resistance also in 95 by Calgene, Roundup Ready in case of Monsanto in 96. So in this way, it started, the era has started in 95 in USA. Thank you for listening. Thanks a lot.